my heart, and it's something we have invested extensively in the past. Okay, let me move things around a little bit. All right. So I hope you can see my screen now.
Yes, we can.
Okay, perfect. Okay, so I'll move. This is mostly an introduction. I know there are many folks very familiar with this topic in VICC, but I wanted to bring also some things that they might have been more research level recently, but as I have here in this slide, you know, we're going faster into the whole genome sequencing era. You know, it's right on in front of our door, and I think it's a very good time. I remember even talks maybe 5 or even 10 years ago that we were discussing that we will reach this point, and there are many challenges, and usually the challenges revolve around, you know, data will be very big, computational resources and analysis, but these, you know, today are definitely much more tractable than they were 5 or 10 years ago. But still, in terms of the interpretation of the 98+2% of our genome, you know, the very large non-coding part, it still is a very large challenge. And even though the germline community is producing whole genome sequencing data in very, very fast pace, and there are an increasing amount of tools, annotations, databases, and resources, the tumor whole genome sequencing arm of this effort is definitely smaller. I think based on, you know, what we see published in the repositories, potentially for every one tumor whole genome sequencing dataset being public, it could be maybe even 2 orders of magnitude more being produced for germline, and that creates also a discrepancy in terms of the maturity across the field. So the interpretation is definitely a challenge. Non-coding, regulatory variants, structural alterations, rare variants, readouts and power. You have a much wider space. So wider space means more things for you to evaluate, and that reduces your power. And there is a lack of resources in the somatic space, as I mentioned, compared to the germline. I will focus more on this part. So there is also a bit of a lag especially compared to coding for nomenclature and standards, which I think is VICC is very well positioned to take a leadership position in this and facilitate the domain and provide the best practices, provide at least seed the discussion, and the community can take it from there. So there are many ways to categorize non-coding variants. I have provided just a few. There are many different ways, and if you look in the literature, depending on you know from which field you're coming from, you will have a preference. So one very common one is transcribed and non-transcribed. Transcribed is usually genic, but that's also not a very good definition. You know, non-coding RNA is intronic 5', 3'. But this is not a perfect definition because promoters are often transcribed, enhancers are often transcribed, and other regions of the genome are often transcribed. And that transcription event is either important for their function or the product is also functional. So that makes things a little bit more complex if you go with this definition. Intergenic and other chromatin architecture elements. So I think I have a couple of slides that I really like to discuss within the lab that show how did we get here? What are the main reasons why this field is quite far behind, apart from the, of course, technology part. This is a publication a few years ago. It's not, you know, 1,000 years ago that showed using public data that most of the mutations happen, for instance, in 3 UTRs, which is our very big preference in the lab to work on, that almost all the mutations, the somatic mutations in cancer in 3' UTRs happen very proximal to the coding region, which makes absolutely no sense, but you know, this is what it is. But if you actually go to the data analyzed in this publication, and it's one of the very few that have analyzed, somatic variants, is that they used whole exome sequencing data from TCGA. And if you actually go to the TCGA panels used, none of these panels that have been used across the TCGA consortium actually captures 3' UTRs. So of course, everything that has been captured is spillover from the whole exome genes. And these small things show where we currently are. Whole genome sequencing is very rare in tumor samples, apart from like ICGC, PCOG, and for now, there are very large efforts that have started, like the Argo, and that are going to change this. But right here and now, you have very few whole genome sequencing data sets, and most of the panels and whole exome probes used, at least in the major studies and cohorts, do not capture any non-coding regions almost at all. So, so it is a lack of available data, which of course makes it more complicated, but also is that non-coding variants have multiple effects. It's not just loss of function or loss or change of the function of the protein. You have a lot of potential regulatory effects. These are mostly the ones that we care about. I think they're practically all. And you have effects on chromatin architecture, transcriptional activity, whether a gene will be transcribed at all or even how much. Post-transcriptional gene expression regulation, which is at least my group's main research focus, and we have invested a lot of resources in this. It's most probably 3' UTR guided, and we're talking mostly about microRNAs or RNA binding proteins, but also translation rate, localization, function of non-coding RNAs, RNA, DNA, RNA protein, and DNA protein interactions, RNA processing such as polyadenylation, splicing, RNA stability, RNA half-life, and methylation, acetylation, histone modifications. Non-coding variants can do a lot of things, which make it much more complicated to track. And are they important? Some have been identified to phenocopy loss of function. That's what I usually mention. Others are just dosage, how much of that gene of interest will be expressed, or whether it will end up in the right place, or whether it will interact with the right interactors. So yes, they might have very important functional phenotypes, but it is a challenge to identify those. So some examples in the literature. First of all, there are not many. The literature is not that it's bursting to it seems with studies examining somatic variants, somatic non-coding variants, because mostly of the lack of available data for people to mine. This is changing. PCOG was the first good example, but there are more studies coming, and it is a reality. It is something that is coming, and we have to prepare. So the available literature mentions a few examples in terms of drug toxicity, progression-free and overall survival, metastasis risk, and response to therapy, which is exactly what we want our variants, to show us and help clinical decision-making. But of course, they're not that well captured. Non-coding drivers are very rare from PCOG. They have shown that it's potentially 1 order of magnitude fewer compared to coding. Of course, the authors mentioned that there's also a lack of power, but I don't think it is. At least for PCOG, what they have, but of course it can be cancer specific and so forth. But at least in this pan-cancer effort, many were identified in the third promoter, which makes sense. In other promoters, and in 3' UTRs, and in other regions that affect genes with relatively straightforward function. But I'm pretty sure there are many more to be discovered. Structural alterations have been a little bit more studied, to be honest, because there are examples in the literature of promoters and enhancers being, for instance, duplicated or truncated and having effects on important oncogenes. And 3' UTR truncations, which is something that we're working on extensively. They're present in diverse tumor types, and they affect how these genes are regulated. So what do you do? How do you tackle them? Are they so exotic as they seem? Practically no. Their discovery is practically the same as coding variants. Either your panel picks them up, especially if your panel, there are some panels, especially for BRCA1 and 2, that have introns and promoters in some groups. Whole exome sequencing, there are some panels like the Agilent version 6 plus UTR that has 5' and 3'. There are other clinical whole exomes that might have promoters or introns in some genes to help for copy number. But the main tool that will substitute all these approaches is WGS. But this is where the similarities end and I think there is a very big benefit when studying non-coding variants, because their investigation might be different, but we have easy readouts to follow. While for loss of function or missense variants that might affect function, you need very specific and specialized experiments that are one protein, one experiment to do with one variant. But for non-coding variants, we have better tools to perform high throughput validation of dozens or hundreds or thousands of variants in one go. But we need different best practices. Everything is different. Even the viewer you use when you're doing regulatory variants, you need additional layers and additional track of information, not just conservation or for instance, presence in GNOMAD. Effect prediction, it can be regulatory, and it can also be multidimensional. It doesn't mean that if it's in a specific region, the promoter region, that it will just affect transcription rate. It might affect other things at the same time. The functional evaluation, there are multiple experiments. Unfortunately, there isn't one single experiment that you can do to capture all potential events, but you have a lot of experiments to work with. And the clinical significance, this is, terra incognita. It's an unknown territory, how to approach this. And I think this is one of the challenges that VICC could tackle. This is an example of a viewer that has actually been used and designed specifically for non-coding variants. It's for pediatric cancers, but it's practically like UCSC genome browser. You have your variants, you have gene expression, splicing, epigenetic modifications. So you have many more things and more tracks than you would expect in your standard variant annotation platform. And it was used, at least in this paper, to provide 2-3 examples. But this is actually how you really approach it. They used it and they found splice mutations, or they found other, an exon skipping event, or for instance, a promoter duplication event, or an enhancer duplication event, and so forth. So you need tracks like these to give you regulatory context in the presence or absence of your variant in order to be able to tackle it. So we also have really, really good tools for in silico methods, because these in silico tools are not created in vacuum. They're created using a lot of high throughput data that I will mention, or even manually curated results. So in silico methods are quite powerful in this domain, not in all subsets, because we have a lot of potential effects, and there are different in silico tools used, for instance, for transcription start site strength, and transcriptional activity. there are tools that you can use to predict this. Translation initiation strength, coding potential, upstream ORF strength, RNA processing, splicing site prediction. These are used currently, these are mainstream even for in our context of coding evaluation of variants, even though some intronic variants can affect splicing, and they might not be coding. poly-A tail site prediction, RNA stability, RNA-RNA interactions, RNA-DNA interactions. This is a little bit behind just because the data that can support it are not as mature, and protein DNA interactions like transcription factor binding site disruption. So there are a lot of in silico tools were quite mature here to capture this, but I think the main benefit of the non-coding variant field is that there are exceptional high throughput experiments that can capture, of course, expression, RNA processing, polydenylation, splicing, and so forth, translation rate or translation events, epigenetic modifications, RNA protein, RNA DNA, and RNA-RNA interactions, even secondary structure through an experiment. There are massively parallel reporter assays where you can plug in hundreds of or even thousands of variants and evaluate whether your promoter is affected, your enhancer is affected, or your 3' or 5' UTR are affected. This is something we use in the lab. And then integrative approaches that take a couple of these at the same time, like RNA expression plus the presence or absence of mutations, and something else, and you're doing a quantitative trait locus analysis or other multidimensional approaches. This is one of our major workhorses in the lab. This is under review. It's fine to share. So we create, for instance, regions where they have the QTL of interest, and we plug it in a gene, and then you transfect with a library of hundreds or even thousands of these constructs with the reference and the alternate allele in your cell culture, and you sequence, and then you quantify how much you have of this and how much you have of this, and they should be 1-to-1 roughly, because that's how much you put, but it isn't. That you see here, these are the results in 2 cell lines, and it's quite surprising that the same mutation have the same effect in 2 different cell lines, which is great. And you have a 2-log fold change. So most of those are not super strong, like it's a 2-fold increase or decrease, which is not small, but it's not 100-time increase or decrease. And this is how MPRA or EMPERA, as other folks call them, assays work. But we have good tools. You can do hundreds or even thousands, and many of these experiments, you don't even have to do mutations. If you have the population of interest, you can use it to juxtapose this. We don't have tools like this for coding mutations. You have to do very, very specific assays to understand whether it's a functional or non-event. But we're very much behind in everything else. Even reporting is a challenge. The nomenclature is quite diverse. Usually it's just one specific variant because many studies do not go in such a detail. Even in whole genome sequencing of tumor sample settings, non-coding variants are omitted completely. We usually do WGS as an even whole exome that gives us good CNVs as well. Or they're just automatically placed in the VUS bin, even though there are publications in cohort studies that have captured them as important. So there is no effort in trying to start at least mentioning what we know for those, even if they're VUSs as well. There was a very recent effort published a couple of months ago in Genome Medicine as the first recommendations for the clinical interpretation of germline variants by a group of colleagues from UK, which is fantastic. Of course, we know there is a very large divide between coding and uncoding clinical interpretation, but at least it shows that we're not alone thinking about this. This is something that is very much in people's heads. But as I mentioned, the germline field, I think it was very long due. We're talking about, I don't know, hundreds of thousands of whole genomes already in the public domain, and large initiatives are going into more than 1 million genomes. So for germline, I think this has been quite long due to be, for their clinical interpretation and start putting some organization in this grassroots kind of self-organized domain. Locus annotation is also a challenge. You have different levels of maturity for non-coding loci. UTRs are rather well defined. Of course, in specific cell types or in specific cases, they might not be the ones we expect, but their majority, I think, are in good shape. Unfortunately, promoters, enhancers, and silencers, another loci for regulation, there is no strict definition that it starts from here and it ends there, and that's exactly where it is. So your variant belongs or it does not belong there. There are some regions that are warmer, that most probably it is there, but it is a gradient. Non-coding RNAs, a big chunk of them super well defined, but the list is growing, and depending who you ask, it's for human genome, it can be in the tens of thousands or more than 100,000. So that is another challenge. And the annotation of a coding transcript or non-coding transcript or a regulatory region, they often overlap. And there is a question in the field, is the coding product always a priority, even if it's not expressed in this tissue at all? Or even if you have, for instance, a very important regulatory locus and a gene of unknown function, which one is the lead you should follow up as being the functional readout of this mutation or event. So when you're reporting functional impact, as I mentioned, we have great experiments, and these great experiments are not made in a vacuum. When you're doing, for instance, the interaction of a protein with a receptor with a ligand, it's a little bit more well defined. But when you're reading out, for instance, gene expression or regulatory events, they are cell type specific or tissue specific. So when you're saying, yes, this variant affects the expression of gene X, there is always a cell or tissue context. It happens in cells X. So, but it doesn't happen in all the others, for instance, or it happens in almost every cell type. Which is the tissue of interest? Does it have to be in the tumor that we're talking about? Let's say, for instance, for gastric cancer, if this is a variant that affects gene expression and we have found it in healthy stomach tissue or in other gastric epithelial cells, is it functional and important in tumor? Does it have to be in tumor? And it doesn't always work in for all the patients. So this is it's a little bit more complicated, and so functional does not mean clinically relevant. That is also a very big divide. We have excellent tools to capture the specificity of an event and the outcome of an event and the phenotype of an event. But whether a twofold increase of a gene expression or decrease, whether that can inform clinical decision making, it cannot be done in a high throughput way. We need still cohorts. We still need the same process potentially that we do with coding mutations. So, but there are currently no guidelines. There are no clear-cut criteria. There is no categorization, and there are no guidelines to at least capture them and report them. And if you capture them and report them, other folks can take over and data mine if it's something out of your bandwidth or interest. So even the definition of clinical relevance for gene expression events or regulatory events is a little bit more complicated. So this is a potential, and this is my last slide. I wanted to focus more a little bit on where the field is, but I think if we are able to put together a working group capturing the status quo with numbers, and saying, we have, for instance, in COSMIC, this number of non-coding variants, in TCGA, in the knowledge bases. These are the guidelines for trimline. I mentioned at least one publication I'm aware of. I'm not familiar with others, but there might be the guidelines for coding. At least capture the lay of the land in a way that can be easy for everyone to see in two, for instance, figures in a table, just for as an easy introduction of where we currently are, because that will inform, what resources we should generate to reach where we want to be. And then there are two axes. One is recommendations, for categorization, for clinical evidence, how the coding or germline frameworks, or even germline non-coding frameworks, could be somatic coding, germline coding, or germline non-coding frameworks should be adapted for somatic non-coding variants. And then the other is best practices, and to start pointing to a toolkit, which annotation, if let's say somebody wants to enter tomorrow, there are no clear guidelines for somatic non-coding variant work. There are no clear guidelines, which annotations are good or not good for such work, which toolkits, databases, knowledge bases, or even for knowledge bases that want to capture these events, there are no guidelines what they should be, writing down, or what information they should have. Everybody is a free-for-all kind of situation at the moment. Which are the good data sets you should include. For instance, we know that in populations, there are not so very good data for all populations at the moment. So if you're not having tumor normal matched in your cohort and you're using public resources, there is an overrepresentation of, for instance, white patients, and usually males in the available databases for many of the tumors. So it might be an important regulatory event, but unfortunately, racial disparities and sex disparities in research affect that as well. If you have tumor and normal, for instance, from the same patient, it makes your life much easier. But there are no guidelines what you should be doing. Filtering, do you still go with the same like 5%, 1%, and the filters that are used currently for non-coding variants, and how they should be represented? Let's say you publish or you create a data set or create a resource, how they should be represented. So there are two, they're very close, but one is more, like practical, how should I approach it? And the other is, what are a little bit the more official tools I have in my arsenal. And then the next thing would be potentially a dry run. We have our recommendations. We start with a table of 50 non-coding variants that capture different fields, and then folks try to use the recommendations to evaluate or represent them and see how it goes. And then if we're happy with it, it's then trying to enable the field, and arm them with the framework and best practices that they can try to get. I don't think we will be, in one go, we will be ready to reach maturity. I think the field needs more work and more data, but I think the earlier we start, the easier will be to bring everybody on board and leverage also the data and reporting that will be in a harmonized way, generated following, this framework and guidelines. So this was it for me. More than happy to hear what you think, and whether you find it as exciting, at least as we do, and your thoughts on this. Thank you.
Thanks for that overview. That was very comprehensive, and it seems like there's a ton to do. I'm really glad that you've got kind of this workflow of steps to take, starting with kind of this, requirements gathering from the community. I think that's a really good, solid first step. Have there been any initial works that we can look at, or parallel works we can look to help guide those activities?
The most comprehensive, but it's germline, is the work that that group did for germline non-coding variants. Of course, there is a big divide conceptually and practically between germline and somatic, but at least I think they did a very nice job, to be honest. But for somatic, you need also the clinical actionability steps afterwards, which is a little bit more complicated, but that is one part. There are research level works like PCOG or other domains, but there is nothing concrete, to be honest, that you can leverage, at least to my knowledge. I think the most comprehensive approach is the germline publication that was published a few months ago.
And the
What
goal.
about anything from the Genomics England group that they had this paper talking about mutation signatures in tumors from whole genome data that had quite a few tumors that were sequenced, but I don't know if I've seen much other analysis of that whole genome data.
Well, there are research level things. There are many, like research grade things. There are many. There is from whole genome. There is PCOG. We're now publishing the project that I showed a couple of slides. It's under review. We'll do a very large, like 3' UTR annotation of non-coding variants. Splicing, there are many groups that have done work in splicing, but all of this, there is a lot of research data and research efforts, but there are almost, at least for somatic, there are no standards efforts. I'm distinguishing between these two because research efforts are usually organic and everybody's doing the best they can, to just get the paper out and you have the data and I'm representing my findings X and I'm annotating with this way, and you're, even with the same data set, you're finding completely different things because you're annotating differently and representing differently. But for standards, only for germline, there is this single effort. But yes, consortia, there are currently, PCOG is one, Genome England is one, Argo has not provided any data, but I think it will be very big. There are some whole genome sequencing efforts in specific tumor types, but usually those only have whole genome sequencing from the tumors, and they don't have functional readouts because usually in this field, it's not enough to capture the variant. You usually need also something to correlate it with, like a functional phenotype. So there are not that many efforts that have the funds to do WGS plus a couple of other dimensions at the same time.
Hartwig, I guess, is another one. I agree. Yeah, I agree with you about the sort of research versus sort of clinical interpretation being very kind of different problems. But those groups might be interesting people to reach out to maybe be part of the conversation about what standards or guidelines like are needed or what they might look like, or yeah.
I had a discussion with Rainbay maybe a couple of years before, when meetings were still in person, who was the first author of the PCOG, for instance, paper for non-coding variants. And still everything is grassroots, but that was a couple of years ago. Potentially, if we reach out again to the community with this in mind, things might be different in how they work. But still, there is no effort to bring these things together, at least try to harmonize the field. These are the IC papers, when you're reporting single cell data or spatial data, these are the things you should have to help everybody, work, or for, as I mentioned, the germline variants, but there is no such, even, what you should be reporting and how you should be addressing this, or what is best practice and what is not for somatic non-coding variants.
I think I saw that Obi had a question. Obi, do you want to ask now, or did they get addressed?
Yeah, I think it was addressed.
Yeah, one part that I briefly mentioned is that for structural non-coding variants, non-coding alterations, there is a little bit more clinical actionability. We know, for instance, for promoters or enhancers, there are some groups that take them into account, like duplication events, for very like MIC or TERT, or like there are folks that actually include this. Splicing variants, even if it's in the intron, but it's predicted to disturb like splicing or do an exon skipping or something like that, even if it's not coding, folks take it into account in their molecular diagnostics. So there are some domains with much higher maturity, which I think they can spearhead this effort. There is proven clinical relevance, and we know this is important. Potentially the others will be important as well. And we know, for instance, that there are events that phenocopy loss of function, even though they're non-coding. The cells, at least on the cellular level or in vivo in the mice, you see exactly the same phenotype as you see if you knocked out the protein completely. So for some non-coding variants is a no-brainer that this is very important. But for all the others where you have like 50% increase, twofold increase, the field needs some guidance. And to be honest, I don't have it. Even if they ask me, what threshold should I be using, I can tell them what we use, but it doesn't mean that it works for all the domains and for everybody at the same time.
I'm wondering how much you want to break this problem down, like, because it's such a large and disparate set of problems, like you might have very different best practices or guidelines for, some of the examples like splicing variants versus structural variants versus UTR variants, or do you want to try and come up with one set of best practices or guidelines to rule them all? Like, is it better to actually break it down into more modular pieces or to try and tackle it holistically?
I think you need a holistic description of your broken down best practices. So your best practices have to be broken down. There is no other option, but at least there should be all of them together so people can see them, side by side. You have one place to look for all the guidelines, but the guidelines, they have to be different depending on what these variants are supposed to do or where they are. I don't think you can escape that.
Yeah. So maybe you have like a kind of umbrella workflow that says if you have a splicing variant, go here. If you have a non-coding structural variant, go there, and so forth. But yeah,
and you can also group them, like variants that affect how much transcription or abundance of your RNA molecule you have. So there might be also two or three different types of variants that can be grouped together, and they can be tackled together in terms of how they're reported or whatnot. But each of them might have different experimental ways to validate or different guidelines on how you should be doing this. But the clinical relevance or they could fall in the same bucket. So it could be also hierarchical kind of approach. All of those can be tackled similarly. All these are singletons. Splicing, for instance, is something that that is its own thing. But there are different mutational events that affect, for instance, gene expression or the abundance of RNA molecules. So that, for instance, is they could potentially be grouped after some level of abstraction and above.
Yeah, the question of like prioritization and is interesting, and some of these things are not mutually exclusive, which also leads to some interesting like interpretations where you have, there's like examples of splicing variants where you actually get two things happening at once in the same cell. Sometimes the variant is actually spliced in, and then in that context, it's an amino acid change of some possible significance to the protein. In other cases, that variant is causing a different splice site to be used where it's actually not there, and the protein is actually quite a different sequence. And so what do you do when multiple things are happening?
Yeah, I keep to be honest, my bar of success very, very low. I think a big success would be just enough to jumpstart a discussion and put the field, interested in organizing or self-organizing or rallying around an effort to get at least homogenized as much as possible. Or this is important, we are already in the WGS era and we're doing it, so let's at least do the best we can to do it. I think the first iteration, if we manage to do that, we are VICC, this is how we see this thing. It is important. We can't avoid it. We cannot, even if we look away, it is there and it's looking at us. We have to tackle it. These are our thoughts on it. And folks might come out and say, Vic was right in 7 out of 10, but in these 3, we suggest more stringent or more relaxed or whatever. Sure. I think getting people interested to at least discuss it, I think that will be a milestone for the field.
So great discussion. I'm Samanthi, new to the group, but I have, so I've been not directly involved with this whole exome sequencing effort at MSK, but I know the group and Mike Walsh in Clinical Genetic Services, whom I work with, and he's a pediatrics oncologist. So he and Yung Khan's group have published recently the whole exome sequencing, clinical whole exome sequencing for the young adults and peds. What in their experience, and I kind of like went through some of those annotation presentations, is for the whole exome sequencing, especially the non-coding enhancer region, they rely a lot on the RNA-seq data. So germline, so this is only 200 samples, but they have a matched tumor normal, but it was not enough to kind of like call any variance significant. I mean, like there's so many singletons, so it's not easy. So what they took an approach is also a matched RNA-seq. So you can imagine it's a extremely intense and expensive, I would say. It's 200 samples, a whole exome, tumor normal, and the RNA-seq. And that's where they could identify some few of, like, if you go through, the paper is out in May of 2022. And you can see that it is like enhancers are still, and the non-coding region is still difficult. Structural variants, on the other hand, as you said, when you have a germline and matched germline, it is, I mean, still doable. But for the non-coding, it seems to me that I can also circle back and I can get more on Michael Walsh, the people who have involved in the annotation, to get their input exactly what were the challenges, but that's what I heard. So RNA-seq, how do we kind of expand the RNA-seq into this equation of annotation is always the biggest question.
These are all excellent points, and I think it is and it isn't the field's fault, because the lack of resources that are available for coding variants. When you have a coding variant, you have a missense, right? You don't go and do a binding experiment yourself to see whether, so you're in your patients, you always have, for instance, binding affinity experiments and functional experiments. You don't. You have resources that you can go and tap into and say this variant affects the binding site of this or the binding domain, and it is deleterious and does this and does that, which unfortunately these resources do not exist. So we're trying to tackle it ad hoc, and we're spending our own resources when we're doing our patients, to get RNA-seq and get a little bit of this and get a little bit of that, just because centralized, warehouses of this information are not easy to tap into. So that's why I'm saying it's not the field's fault because these data are not there, but it is the field's fault because it never pushed for these resources to be as readily available. My group is spending, an R01's worth funds just for 3' UTRs, and it's not enough. So you need bigger things than a single group can tackle. As you mentioned, to even 200 whole genomes or whatever is with RNA, it's a lot it's a huge effort for a single group. So I think just I think sounding the bell that as we're doing things, it will take us forever. And we need to put this as a priority. And for all the folks that are, for instance, doing whole genome sequencing, they're actually now clinical and CLIA certified for tumor. And for some tumors, for instance, there are publications that show with WGS, you capture whatever you wanted with your whole exome plus more. So there is a push to move towards one uniform platform. But we need to point that we're not only we're not ready, but at least, you know, if you are to start, these are at least some guidelines. And these guidelines did not come from a single group. They come from a group of people that actually sat down and discussed it and reached the consensus that it's not perfect, but at least if we were to start, that's how we would have approached it. And then the community can refine it further. You can get feedback. You can have versions of this, but I think it will be at least your starting point for this, and it will be organized and not, you know, everybody has their own opinion.
So is there any possibility we can think of, like I work in the germline space, so I'm just thinking of like, and which I'm not able to understand, we can brainstorm here, is like the GTEx, which is a tissue panel RNA-seq reference panel. So that we use heavily depend on when we do a germline annotation. So kind of like building a resource as a reference. But here we don't know because the cancer types would be different for like, and it may have a different. So like my question is like, even I'm not able to trying to understand how would we even build a reference genome, like reference cohort, so that not every time we need to do an RNA-seq, but we can impute the sequencing and understand the variant interpretation. So
no, you're absolutely right. Of course, it's not VICC's job to do that. That's way beyond our, you know, our capabilities and agenda. But VICC could point to what resources could help. And I think PCOG is already doing it. You know, they're now doing WGS on TCGA samples, and TCGA has everything under the sun for these samples. You know, from imaging to H&E to epigenetics to RNA, small RNAs, everything. So when you reach 12,000, you know, now I think it's like 3,000, but when you reach 12,000, you should be, is it 3,000? I think those that have everything are maybe 1,200. But when you reach 12,000, I'm not going to say you have enough for all the tumors, because for some tumors it's going to be 1,000 plus, and in other tumors it could be 150. But at least there will be something, you know, there will be something there. But at least Vic can suggest, you know, if there are consortia doing this, and that, it would be good to have A, B, C, and D, or just look at how TCGA is doing it. It's almost there. It would just need this and this. So it's not our job to do it, but we can say what we need if we wanted to have a resource like that.
Yeah, that would be great. And I kind of think like that is the resources once, like, I don't know if with 3,000, we would be able to annotation side, we can provide, like we can set up some recommendations and guidelines that, and like, let's say with MS, all these other research teams who have like smaller cohort and kind of combine this like whole recommendation panel kind of thing, like a very initial GWAS era. So how do you prioritize a SNP kind of question, right?
All right, we're at the hour, so we're going to have to pause the discussion here. Thanks so much for the presentation and introduction to this, area of study, Ioannis. If there are any questions, can people reach out to you directly at a given email address?
Yeah, of course.
Okay, great. So we're going to resume, of course, in 2 weeks, or maybe 3 if there are 5 weeks this month. Watch your calendars. We'll see you all next time. Thank you.
Thank you so much.
Thank
Take care,
you.
everybody.
Thank you for a great discussion.